The DNA paternity report shows the results of laboratory tests, which provide evidence regarding whether the alleged father could be the biological parent of the named child. Let's go over the components of a paternity report. The paternity test report has 4 sections: genetic system table, combined paternity index, also called combined direct index, probability of paternity, and test conclusion. The genetic system table contains all the data results in the report. The laboratory tests body fluids or tissue of the mother, if include, child, and alleged father to locate certain pieces of DNA which may differ from person to person. The location of the particular piece of DNA along the entire chain of DNA is called a locus, and the laboratory finds the locus by using a chemical called a primer. The report shows numbers, in the first column of the chart, to indicate each locus that was tested. Our lab tests at least 20 different DNA locations listed in the STR locus column and then compares the data with the same locations for other tested parties. A person's own piece of DNA found at a particular locus in a paternity test is called an allele, and may also be referred to as a genetic marker. Each person has 2 alleles at each locus. The number associated with each allele represents the length of the DNA segment. And often they are not the same size. If only one number is given at a locus, then both alleles at that locus are the same size. For the child's 2 alleles, one allele comes from the person's mother, and the other allele comes from the father. If, for example, a child has 2 alleles that are sizes 11 and 15, and if the mother has sizes 5 and 15, then the child inherited the size 15 allele from the mother. The child has to have inherited the size 11 allele from the father. The size 11 allele is the obligate paternal allele. Generally, the alleged father must have an allele this size if he is the biological father of the child. The box in the chart on the laboratory report highlights the obligate paternal allele and the alleged father's matching allele, if any. If the alleged father does not have the matching allele, then he usually cannot be the biological parent. If he does have it, then he could be the father.
A paternity index, or PI, or direct index, or DI is calculated based on information including the portion of the male population that has this size allele. This index is included in the PI or DI column for each DNA locus. A combined paternity index or combined direct index for all of the tested alleles is then calculated and written in bold type below the chart, along with the probability of paternity. The probability of paternity is the percentage likelihood that a man with the alleles of the alleged father is the biological parent of the child, as compared to an untested, unrelated man of the same race. For a long time, the standard practice in the industry for establishing paternity has relied on examining 16 STR loci. However, at our laboratories, we go above and beyond by testing 22 STR loci, ensuring a level of accuracy that is unparalleled. Our tests are designed to meet both CODI's and European testing standards, guaranteeing the highest level of discrimination among all autosomal STR kits available. In general, if the tested father is confirmed to be the biological father of the child, the result is a positive paternity inclusion. In such cases, the probability of paternity will exceed 99.9%. The statement of results in the report will state, "The alleged father cannot be excluded as the biological father of the tested child." Based on the analysis of STR loci listed above, the probability of paternity is 99.9999%. Conversely, if the tested father is not the child's biological father, the results will be exclusion of paternity. The probability of paternity in this case would be 0%, and the statement of results on the report will read, "The alleged father is excluded as the biological father of the tested child." This conclusion is based on the non-matching alleles observed at the STR loci listed above with ADI equal to 0. the probability of paternity is 0%.